There are four different pathways through which the proteins are transported from the plant cell cytosol to the thylakoid lumen. The pH-dependent pathway is one of them. Let us discuss about it. The cytosolic metal binding precursor protein has a carboxyl terminal domain and an amino terminal domain. The amino terminal domain has a stromal import sequence and a thylakoid targeting sequence. The stromal import sequence directs the import of precursor protein from the cytosol to the stroma through TOC complex present in the outer membrane of thylakoid. Now, the metal binding precursor protein reaches the intermembrane space. From here, the precursor protein is then imported into the stroma of thylakoid through TIC complex, which is present in the inner membrane of thylakoid. Now the metal binding precursor protein reaches to the stroma. Inside stroma, the metal binding precursor protein undergoes cleavage to produce a metal binding protein and a cleaved stromal import sequence. Due to this cleavage, the thylakoid targeting sequence in the metal binding protein is exposed. The cleaved stromal import sequence remains in the stroma. During the first step of delta pH pathway or pH dependent pathway, the metal binding protein undergoes folding to produce a folded metal binding protein. The metal ions present in the stroma then binds to the folded metal binding protein. The metal ion acts as complex redox cofactors. The RR sequence present at the terminal point of thylakoid targeting sequence of folded metal binding protein represents two arginine amino acids. These two arginine amino acids and a pH gradient across the inner membrane are required for the transport of folded metal binding protein with bound metal ion from the stroma to the thylakoid lumen. As these two things are available, the folded metal binding protein with its bound metal ions is transported from the stroma to the thylakoid lumen through a translocon present in the thylakoid membrane. The translocon is composed of at least 4 proteins related to proteins in the bacterial inner membrane. In the thylakoid lumen, the folded metal binding protein with its bound metal ions undergoes cleavage to produce mature metal binding protein and a cleaved thylakoid targeting sequence. Please like, subscribe and share.